Thanks, Rick. A bit of a change of pace here, but hopefully interesting. So you may ask, is CLL familial at all? What evidence do we have that it is? And there are a number of lines of evidence in support of this. Somewhere between 10 and 12% of CLL patients have been reported in the literature to have a first-degree relative with a lymphoproliferative disorder, which could include CLL or another lymphoma, although in about 2/3 of cases, the other disease is CLL. There are rare families reported with apparent Mendelian inheritance of CLL. And the population attributable risk due to inherited susceptibility is 35%, which is actually quite high, making CLL one of the more heritable cancers. This is a sample pedigree from a family enrolled on my tissue banking study, which we'll come back to later, but you can see 5 of 11 individuals have CLL in the second generation, and in the bottom generation, 3 out of 5 have CLL. Population-based studies from Scandinavian registries have found that the increased risk of developing CLL to a first-degree relative of an individual with CLL is 7.5-fold. And you can see here that's a higher number than for any of the other related lymphomas. So given these data, about a decade ago, we started systematically screening all of our new patients seen in the CLL lymphoma program at Dana-Farber for a family history of CLL, with a one-page IRB-approved self-administered questionnaire. Our response rate was about 71%, and we also reviewed physician notes. In the first 2¼ odd years, we had 1948 evaluable patients, and to date, we've screened over 6,000. Those with a family history are eligible to then enroll on our tissue banking and natural history study. This slide summarizes the malignancies reported in first-degree relatives of the probands with various lymphomas, including CLL. And you'll note that in general, the incidence of these cancers looks high, but that in particular, lymphoid cancers appear to be enriched, particularly in the CLL group, almost as frequent as breast cancer. Here you'll see that amongst our patients, 13.3% of CLL patients reported a first-degree relative, namely a parent, sibling, or child, who also had CLL or lymphoma. And 17.5%, almost 1 in 5, reported a first or second-degree relative with a lymphoproliferative disorder. The estimated lifetime risks of developing CLL if your mother or father had CLL in our data set were 2 to 3-fold. And so we're also interested in looking at what lymphomas these individuals developed within their families. And you'll note that most commonly, it was the same lymphoma that was seen in the relatives. But that particularly in the CLL group, there's also a significant incidence of non-Hodgkin's lymphoma, making some argument that it's possible that there are multiple phenotypes. Some families may have just CLL, and other families may have a range of different lymphomas. I wanted to say a word about monoclonal B-cell lymphocytosis. So as you know, CLL phenotype clones have been described to increase with age in normal individuals when measured by highly sensitive flow cytometry. They occur in about 3% of individuals over 40 and about 5% of individuals over 60. And this led to a new diagnostic entity, MBL, or monoclonal B-cell lymphocytosis, which is the situation of circulating clonal B cells, usually but not always of CLL phenotype, with an absolute B cell count less than 5,000 and no other features of a lymphoproliferative disorder. So those latter two features mean that these individuals do not meet criteria for having CLL, but nonetheless have this population of cells. And studies have suggested that all patients with CLL do have a pre-existent MBL, but that the prevalence of MBL is several hundredfold more than CLL, so that most MBL does not develop into CLL. So you may think that MBL would be more common amongst relatives in CLL families, and that appears to be the case in that it's been reported in 14 to 18% of first-degree relatives in CLL families in several studies. And the relative risk is higher at younger ages when it's relatively rarer in the general population. And so this may be a marker for the at-risk phenotype for developing a lymphoproliferative disease. So what exactly is familial CLL? Well, it's traditionally defined as at least 2 first-degree family members with CLL, although I would argue that it should possibly be expanded to include other lymphomas as well. Now, the disease features appear largely similar to sporadic CLL based on the literature that's been published. There's no difference in stage or need for therapy. No difference in 10-year overall survival. Onset has been variably reported to be about 2 to 10 years younger in individuals with a family history, but there's no evidence for anticipation, which is the genetic phenomenon where subsequent generations develop the disease at a younger age than the parental generation. We looked at familial CLL in the CLL Research Consortium database at a time when there were 3,328 patients that we could evaluate. And we broke the patients down into 4 groups based on family history. 14% had a family history of CLL, 13% a family history of other hematologic malignancy, including lymphoma, 43% a family history of non-hematologic cancer, and 30% were sporadic with no family history of any cancer. Mean age at diagnosis was basically similar amongst these groups. There was a possible trend toward 1 year earlier in the family history of CLL group, which is not likely to be meaningful, even if real. And you'll note that the age at diagnosis is quite young because the members of the CLL Research Consortium are generally tertiary referral centers. No differences were observed between groups in rise stage, beta-2 microglobulin, IgG levels, CD23 or CD38 or ZAP70 expression. There's no difference in the incidence of mutated IgHV compared to unmutated across groups. And there was also no difference in the VH gene usage across groups. So, in summary, there's little evidence for any biologic differences between familial and sporadic CLL, but cytogenetics have not really been adequately investigated. In a small study, we found a lower frequency of 11q deletion in familial CLL, but it may have been subject to bias due to the size of the study and really would need to be confirmed. And a large-scale analysis of clinical outcomes in comparison to sporadic CLL is also needed. And we do hope to do these things in the CLL Research Consortium, but have not yet been able to, or currently resuming that effort. So how do we think about finding genes for familial CLL? And this figure comes from a review written by Richard Holston a number of years ago for thinking about models of genetic causation based on the number of lymphoma or CLL cases in a family, ranging from 1 to 4. So if you consider if there's only a single case in a family, that has a high chance of just being sporadic and not genetically related, and is very unlikely to be related to a very high-risk allele contributing to the disease. As we move from 1 up to 4 cases in a family, as you might expect, the chance that this is random and not genetic goes down substantially. The chance that it's due to a high-risk allele goes up substantially. And then the low-risk alleles that confer some moderate risk are somewhat scattered throughout. So what are the implications of this? Well, so a very high-risk allele, conferring, for example, a 25-fold risk of the disease, would be infrequent in the population, and you might only need a couple of alleles to cause the disease. This makes these relatively easy to find, similar to Mendelian inheritance. If we go down to the bottom line, where we're looking at a relative risk of the allele of about 1.5-fold for developing the disease, and this is on the order of what you find in genome-wide association studies, where relative risks tend to be on the order of 1.2 to 1.5-fold. Those types of alleles have to be much more frequent in the population, may be found in as many as 10% of the population. But the implications of that are that you need several hundred to cause the disease, making it much harder to identify these and requiring potentially up to tens of thousands of individuals to be studied. So what approaches have been used? So traditionally, of course, linkage, as well as candidate gene studies, including both direct sequencing and association studies. And then more recently, genomic approaches, including genome-wide association studies, and we're interested in detailed genomic analyses of familial versus sporadic CLL. So linkage in CLL has been troubled by the fact that early studies were underpowered. The largest study included 206 families, where most of the families had 2 or 3 affected individuals. The strongest signal was at 2q21 at the CXCR4 gene. And follow-up studies, however, showed no association between SNPs and CXCR4 and CLL. There were 3 mutations in 186 familial CLLs in CXCR4 and none in 213 controls. But I haven't seen any further follow-up characterization of that observation. Another very elegant study looked at the DAPK kinase gene. This was from Ohio State. They studied a family with Mendelian inheritance of the disease and identified a SNP allele that affected expression of the gene that segregated with disease in this one particular family. However, it was not seen in 358 other CLLs. And in general, this has been the message from candidate gene and linkage studies, that they've primarily identified private mutations in individual families, including DAP kinase, as well as 2 mutations in the miR15-16 microRNA at 13q, which have been reported by Carlo Croce. Candidate gene studies have found no significant involvement of HLA, ATM, or a variety of promoter SNPs in apoptosis-related genes. So that leads us to genome-wide association studies, work which has been primarily done by the UK group led by Holston, as well as the Mayo group with Susan Slager. And there have been several studies. And I would remind you that a genome-wide association study identifies an allele, a SNP allele, which is highly statistically associated with getting the disease, but typically is not in the coding region of a gene, and how it works biologically or functionally is usually unclear. And so I highlighted here a couple where we see a potential biologic significance at 6p25 for the IRF4 gene, which I'll talk more about later. And then an allele at 8q24, which is in a region that serves as an enhancer for MYC, which has been associated with the risk of many solid tumors as well. Then the IRF8 gene. Interestingly, many of these have been tested and validated in monoclonal B-cell lymphocytosis as well, suggesting that, in fact, the risk profile is similar for MBL and CLL. There have been 2 more recent GWASs, one focused on familial CLL that focused particularly on the IRF8 gene as well as the HLA locus. And then a recent meta-analysis of 3,100 CLLs and 7,600 plus controls, which identified a number of new loci with apparent biologic relevance, affecting potentially BCL2, FAS, NOXA, and LEF1, which is highly expressed in CLL. So does this explain familial CLL? Well, the frequency of these GWAS risk alleles has actually not been explored in detail, specifically in familial cases. We had looked at one of them, RS8720771, which is the one located in the 3' untranslated region of the IRF4 gene. And that's homozygous in sporadic CLL and familial CLL at the same rate, 34 to 35%, which is higher than the general population, but not different between familial or sporadic CLL. And so modeling has suggested that these common SNPs identified by GWAS may have the potential to explain up to about half of the risk of familial CLL. But that suggests that we still have half left to explain. And some of that may be from other common loci that have not yet been found. But it's also possible that some of it is from much rarer alleles that are more penetrant, that have a higher relative risk for developing the disease. Or some of it may be from intermediate frequency SNPs that have intermediate effects. So we've been interested in trying to study familial CLL using some of the extremely high-resolution techniques that are now available to define genetic events. And this really requires that normal germline variation be cataloged, which it increasingly is, although that's still a bit of a problem in doing these studies. So can we identify any genetic events underlying familial CLL? We've been comparing probands with familial CLL to those with sporadic CLL, using copy number, gene expression, whole exome sequencing, which is still in progress, and we're moving into whole genome sequencing. And then we're also studying multiple affecteds in families with strong CLL inheritance. So from this, have we seen any differences in somatic genetic events, so those that are acquired as part of the tumor, between familial or sporadic? Basically, the answer is no. Copy number aberrations, such as 13q trisomy 12, are largely similar, with some minor variation that has been seen in small studies. And by sequencing, there are also no apparent differences. In our sequencing studies, somatic mutation rates appear to be similar between familial and sporadic CLL. And when we look at some of those driver mutations, which Dr. Kay presented earlier, between familial and sporadic CLL, there doesn't appear to be any significant difference in their occurrence. So then what about germline events? So we have been able to identify 2 unique copy number variants amongst 44 families with CLL that we've studied with copy number. One has occurred in each of 2 different families that have apparent Mendelian inheritance of CLL, which I'll show you. One is a 500 kilobase loss at 13q14 affecting the DLU7 gene, and another is a 720 kilobase gain of 6p25 affecting the IRF4 gene. So this is the first one. We noted that there were 3 individuals who carried germline deletions, shown in the blue bars, of just 2 genes in 13q, DLU7 and RNase H2B. You'll note that these did not include the DLU2 or MIR15-16 region, which is usually considered to be the minimal region of deletion in the tumors, in the somatic region. And we noted that these 3 individuals corresponded to the pedigree, shown here. 2 affected siblings, and then one of them transmitted this to one of their children, who is as yet unaffected, but also only in her 30s. We did test the unaffecteds with the asterisks for MBL and did not find it in any of them yet. So we're following up on them to find out if this deletion corresponds with disease in that generation. But meanwhile, we were interested in seeing if this behaves like a classic tumor suppressor gene, which actually it does. And so each of the 2 affected individuals carry the loss in 1 copy in their germline, and they lose the 2nd copy in the tumor, which is shown here. One of them loses the exact same region without further extension, and the other loses a larger region that encompasses the more typical 13q loss that we see in CLL. And that's shown also by PCR on the right. We looked further at DLU7 expression in our family as well as CLL in general. And what you can see is that it's low in all CLL compared to normal B cells, regardless of deletion of the region or our family. We resequenced the gene, but didn't find any other somatic or germline mutations, suggesting that expression is or deletion is the way in which this is regulated. And so what about DLU7 as a tumor suppressor in CLL? Is there any other evidence for that? So in the vast majority of CLLs, it is included in the somatic minimally deleted region at 13q. And as we just saw, it's expressed at very low levels essentially in all CLL. And it's been recently reported by Carlo Croce's group to inhibit NF-kappa B activity by inhibiting TACI and BCMA receptors for APRIL. Conditional deletion in B cells in mice results in a lymphoproliferation resembling CLL. So we believe that this family has what appears to be a single allele knockout of this gene. The second germline event is shown here. It's at the extreme end of chromosome 6p, the short arm of chromosome 6, and is a gain of about 720 kilobases. And it's in the family whose pedigree I showed you at the beginning. The gray box shows the individuals in this family who are enrolled in our study. 2 affected individuals carry this gain. Their 2 unaffected siblings do not. And then each of the 2 affecteds transmitted it to one of their 2 children. And similar to the other family, the children are only in their 30s and are as yet unaffected by MBL or CLL. Now, this region encodes 4 protein coding genes, DUSP22, IRF4, XOC2, and HUS1B. Now, IRF4 I mentioned earlier as having been implicated by genome-wide association study. We sequenced all 4 of these genes, as well as the untranslated regions of IRF4, in 90 familial CLLs, found no mutations, no germline polymorphisms. We double-checked the copy number with PCR and found that there are, in fact, 4 copies of this region in the germline of these individuals. And those 4 copies show selective gain of the genome-wide association risk allele, as one might expect. Interestingly, this gain confers lower expression of IRF4 compared to individuals without it who have CLL. Now, this is consistent with the effect of the risk allele seen in genome-wide association study, which also lowers IRF4 levels, even though that's counterintuitive, obviously. And what else do we know about IRF4 and CLL? Well, it's a transcription factor somatically mutated in 1.5% of CLLs. There are rare translocation breakpoints in CLL. Knockout mice show a block in B-cell maturation and develop lymphadenopathy. And there's a very recent report, just pre-published in Blood, of another mouse model with a VH11 mutation in IRF4 knockout, which develops CLL with 100% penetrance. Again, consistent with the fact that low IRF4 levels promote CLL. We do not, however, know how the gain of this region results in lower IRF4 levels. So what about sequencing? So we're really just embarking on this. We have 165 samples that have undergone whole exome sequencing, 60 of which are familial, and we're analyzing the germline for rare high-impact variants, variants enriched in CLL, and variants that have high biologic plausibility, all in comparison to actually tens of thousands of normal controls available at the Broad. We're analyzing paired family members for shared novel variants of likely functional significance. I can't yet show you systematic data from that analysis, but I can tell you a little bit more about the two families with the copy number variants in terms of their genomic profile. So this is family A, which has as its only shared germline event the 13q14 deletion CNV. There were no mutations by whole exome sequencing shared amongst these two affected individuals. And in terms of their disease, it's fairly similar. They both have 13q deletion and relatively stable genome by copy number. They both have a mutation of SF3B1, which is a newly described driver in CLL, as well as by gene expression, activation of Notch and Wnt pathways. And their clinical behavior has been similar, generally indolent, but both have progressed to treatment. In contrast, we have family B, which carries the 6p25 gain. They have 4 shared single base variants by exome sequencing, 1 nonsense event, 1 silent, and 2 missense. And none of those 4 have any obvious relevance to B-cell biology. We're currently trying to sort out whether they are significant or not. Although the nonsense variant sounds promising, all of us carry about 200 nonsense variants that are not of clinical significance. And in terms of the disease in these two individuals, it's quite different. Individual 1A has a very indolent disease, only 1 copy number abnormality, which is 13q deletion, and no driver mutations. And this individual has remained untreated with CLL for over 20 years. Individual 3A, in contrast, has a quite unstable genome with 5 copy number abnormalities, including 17p deletion and 8p deletion. They have a p53 mutation, and this individual succumbed to their refractory disease after about 1.5 years. And so this family makes the point that even a shared underlying genetic predisposition does not necessarily result in the same disease outcome. So what can we say in summary about familial CLL? So first of all, CLL is commonly familial. The phenotypes likely vary, some including only CLL within a family, and others including any form of lymphoma or lymphoproliferative disease. Having monoclonal B-cell lymphocytosis, probably for the purposes of these studies, counts as affected phenotypically, although that is still something that is being validated. Elegant work by Mark Lanasso looking at individuals with MBL in the context of families has found that these individuals can sometimes develop multiple precursor MBL clones that will come up and die off, and then one may take and advance and become CLL. But it suggests that the genetic predisposition is allowing multiple clones to develop over time. Shared genetic predisposition does not mean similar disease prognostic features or behavior. Neither linkage nor association has been particularly fruitful in identifying genes. And in fact, most families with Mendelian type inheritance that have been studied seem to have a private allele affecting a gene that's otherwise involved in the disease. This includes the DAPK kinase gene, MIR1516, as well as the 2 families we've found with the germline copy number variants. So none of these have necessarily provided broader insight into the general heritability of CLL. Common variation from genome-wide association studies, including HLA, contributes perhaps up to half of disease risk, but does not explain the other half. So what is the source of remaining heritability here? It's possible it's very common variants of very low effect, in which case it may be hard to find them unless we can accumulate tens of thousands of individuals to study. But it's also possible that it is rarer, somewhat more penetrant events, which we may therefore be able to identify with sequencing type studies. So we're continuing to mine the germline data for copy number and exome sequencing, comparing it to normal controls, expanding to include whole genome sequencing, RNA-seq, and epigenomic analysis to get information on regulatory regions, microRNAs, epigenetics, and non-coding regions. And the pie in the sky goal is to try to develop a comprehensive genomic description of familial CLL, but you've probably noticed from this talk that that is a very challenging problem. So I'd like to acknowledge my lab, as well as in particular my collaborators, Kathy Wu and Gadi Getz at the Broad Institute, and my funding sources. Thank you.